Hello, everyone. Welcome to today's live broadcast, Breaking Bad with 3D, the development of high-throughput screening platforms to interrogate physiologically relevant models. I am Judy O'Rourke of LabRoots, and I'll be your moderator for today's event. Today's webcast is presented by LabRoots.com, the leading social media site for science professionals, and sponsored by PerkinElmer. PerkinElmer is a global leader focused on improving the health and safety of people and the environment. Their innovative detection, imaging, informatics, and service capabilities, combined with deep market knowledge and expertise, help customers gain greater insights into their science. Before we start, there are a few instructions. We want to hear from you during this interactive broadcast, so please ask questions or leave us a comment. Answers are welcome too. You can do this by hitting the green Q&A button at the lower left of the presentation window and typing in your comments and questions. We'll try to get to as many as we can, and we'll follow up if we don't have time today. Want a better look? You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you can't hear or see this presentation properly, let us know by clicking on the support button on the top right or use the Q&A button. We'll make sure we try to resolve any issues. Now let's get right to today's presenter. We are proud to welcome Madhu Lal-Nag, PhD. Dr. Lal-Nag serves as the head of the Trans NIH RNAi Facility at the National Center for Advancing Translational Sciences, NCATS. The facility is responsible for developing and conducting genome-wide, physiologically relevant phenotypic assays for intramural researchers. She joined NCATS in 2013, where she worked as a research scientist primarily to develop an assay platform of 3D physiologically relevant multi-cell type disease models that are amenable to high-throughput screening. Her PhD in Molecular and Cellular Oncology is from the George Washington University. Dr. Lal-Nag has extensive experience in the miniaturization and optimization of physiologically relevant cell-based 2D and 3D assays to make them amenable for the screening of high-impact, small molecule, and functional genomics libraries. Her complete bio is found on the LabRoots website. I will now turn the presentation over to Dr. Lal-Nag.
Thank you, Judy. Hello, everyone. I'm delighted to be able to talk to you today about paradigm shifts in the development of high-throughput, three-dimensional oncology models for screening with a view to enhancing predictivity in drug design. As reported by the American Cancer Society in 2017, 1.6 million cases of cancer are still reported every year, and more than 600,000 people will die from this disease. So this really necessitates a paradigm shift in the way that we think about not only the disease, but how we're going to mimic this in a plate. If the idea is to go from a plate to a patient, how do we best mimic the disease in a plate? And to do this, we have to take a step back and think about what we are really trying to model. When you talk about cancer, not only do you have multiple types of cancer and multiple stages of cancer, but you also have within each stage, whether you're looking at a primary tumor or whether you're looking at a metastatic disease. And as it were, these are extremely different beasts and have to be treated almost as separate diseases from the perspective of screening models to be able to get to an answer that would actually be predictive and prognostic in human beings. So if you look at the difference between primary tumors and metastatic disease, the difference is location. It's all about the location of the disease. Depending on where the cancer is, the assay must recapitulate that biology to be able to give you a prognostic answer. So if you look at the primary tumor here that's actually encapsulated by the basement membrane, this is designated as stage 1 or 2 of any primary tumor. However, when these cells are able to break the integrity of the basement membrane, extravasate, and go all over the body to establish these micrometastases, if you will, this metastatic disease actually becomes a completely different beast and has to be treated as such. So imagine, if you will, the spotted plant, and you think about this as a primary tumor. You can use a pair of garden shears to actually trim this plant and keep the proliferation of the growth of it under control. But if you use that same pair of garden shears and try to control the growth of this forest, that would be a gargantuan, if not impossible task. Again, this goes back to talking about what are you trying to model, and how do you best recapitulate that in an in vitro plate-based environment. So it all comes down really to understanding your microenvironment. So what you have on the left here are 2 ovarian cancer cells in the milieu of its stromal microenvironment. Now, what has been done a lot extensively in the past has been to usually plate cells in 2-dimensional culture and try and mimic this disease. However, that has been pretty unsuccessful in getting drugs to the market, because what you're really trying to mimic is models which mimic the geometry of these 3-dimensional epithelial cells and how they interact with the microenvironment. Plating them in a 2-dimensional format does not really serve that purpose. So when it comes to primary tumor models, the most frequently asked question is, well, why 3D? The answer to that is pretty straightforward. It turns out that human beings are not 2-dimensional. So how do we now tackle the question of what is the best model to mimic in a plate so that you can get better drugs to the clinic faster for patients? So with that in mind, at NCATS, what we thought was we would start off developing these 3-dimensional models, both for solid tumors as well as metastasis, and then feed them through a robotic system so that you have a much higher success rate of getting compounds into the clinic. Now, if you look at the left side of this, the left panel on the slide, you'll see something that most of you are probably familiar with, 2-dimensional clonogenic survival assays on the left, which are the gold standard in the industry for getting any sort of oncology compound into the animal preclinical model. But what ends up happening is that there's a high attrition rate of compounds that goes into animal models, leading to an even higher attrition rate of compounds getting into the clinic. However, if you start off with a physiologically relevant 3D model and are able to screen thousands, hundreds and thousands of small molecules and do functional genetic screens in these models, the success rate of getting these compounds into clinic is much, much higher with a shorter time to clinical trials, as well as a much less depletion on the resources that are spent on all of these studies in research labs. So we've talked about why 3D, why we need 3D models, but the next question is, what is a 3D microtissue? And I don't think we as a community have actually one answer to this. It really just depends on what you're modeling. So we have 3D tumor spheroids, something that I think many of you are probably very well aware of. You have 3D layered tissues where you're actually trying to build some sort of complexity into your system to mimic what's going on in the body. And then we actually have 3D bioprinting of architectural design tissues to serve as actual tissues and an actual iterative model into which you can spin your small molecules that have gone through all of the other 3D models. The idea really is to develop, benchmark, and implement a series or a platform of 3D models that can then be much more prognostic for a set of small molecules that you send into preclinical studies. With that in mind, today I'd like to first start with focusing on 3D layered tissues with a particular case study that we have using the human ovarian omentum. So this is a project that was started in collaboration with the University of Chicago, and it was the idea was to benchmark this particular assay to make it much more prognostic for women suffering from stage 3 and 4 ovarian cancer. And one of the first collections of small molecules that was used to develop this assay was what we call the MIPE collection here at the National Center for Advancing Translational Sciences. We have over 2,000 compounds that have a diverse and redundant mechanism of action represented. So there are several compounds per target, and this really allows us for a target-based enrichment analysis of the responses that you get for the assay. So given that background, I'll give you a little bit about why we chose to focus on ovarian cancer. As I'm sure many people are well aware, ovarian cancer is the leading cause of death from gynecological malignancies worldwide. The earlier stages of ovarian cancer, so if you go back to the previous slides where you look at the solid tumor when it's stage 1 and 2 and it hasn't breached the integrity of the basement membrane, are actually indistinguishable. There are no obvious symptoms. However, once it's actually breached the integrity of the basement membrane is when you have stage 3 and 4 high-grade serous ovarian cancer, which is usually associated with a late diagnosis and an extremely dire prognosis. As you can see on the panel on the left, between 2005 and 2015, there hasn't been a huge shift in either the mortality or the morbidity associated with ovarian cancer. So this really necessitates, again, a huge change in the way that we seek to understand the tumor and its microenvironment. And this is going to be extremely important for the development of unconventional targeted therapeutics. So we know now that ovarian cancer is aggressive and rarely detected at an early stage. So there is a huge unmet biological need to identify small molecules that we can turn into drugs to target metastatic disease. The objective of this particular 3D assay that we collaborated on with people at the University of Chicago was to identify inhibitors of ovarian cancer cell adhesion to the site of metastasis. So imagine, if you will, the omentum. The omentum is a fat pad that lies immediately below the diaphragm in women and has fibroblasts, mesothelial cells, and adipocytes. What's important about the omentum to remember in the context of the assay is that it is the first site for metastasis in aggressive ovarian cancer. I'm telling you this so you can put into context how we model this in a plate. So in the plate on the left-hand side where you see the blue, the green, and the red cells, those are actually patient cells, the blue and the red are. And then you have this GFP expressing cancer cell line that we look for invasion through this matrix, this three-dimensional matrix, which actually represents the human omentum in a woman suffering from ovarian cancer. And on the right, you have some in vivo data from some compounds that actually came out of the screen in a mouse model, which is our preclinical model, that actually go to show that the results from a three-dimensional in vitro screen were highly predictive for how these compounds would behave in an animal model. So the first thing that we had to do was miniaturize the process. How do you actually recapitulate a human omentum, which has multiple cell types, in a plate? So to do that, we went through many iterations of patient cell lines, and as you can see, one of the first things that you do with high-throughput screening is to look at plate-based QC parameters and make sure that the Z factors are all above 0.5, which essentially tells you that you have a robust and reproducible assay. Now, why this is important in this particular context is because we are actually, instead of using cell lines, using patient cells from multiple different patients. So not only do you have multiple cell types, you have multiple patients that are represented. So there is a reasonable expectation that there'll be a certain amount of variability in this assay. And to our amazement and surprise, really, this assay was extremely robust and reproducible, which really speaks to the fact that once you're able to develop a model that accurately or to the best of our ability mimics human disease, the disease mechanism comes into play and makes the assay extremely robust and reproducible. So after having determined the plate-based QC parameters, we actually optimized a biological control, which you can see on the right, where you have tomatine and DMSO. Tomatine actually very effectively prevents the adhesion of these cancer cells to the omental layer. Once you wash this off, you actually see no cells in the tomatine-treated plate, whereas in the DMSO, which is the non-targeting control plate, you actually do see that these cells are not only able to adhere to the fibroblasts and mesothelial cells, but they're able to migrate to the bottom of the plate, as you would see in metastatic disease. Moving towards in vivo validation, a lot of the small molecule compounds that came out of the screen were actually put into mouse models of metastasis and adhesion against various different cell lines. And as you can see, all of these compounds reproduce beautifully in a mouse model of metastasis and adhesion. Furthermore, not only was it associated with a decreased metastatic load, but it was also associated with an increase in tumor-free progression and overall survival, thereby going to show that once you can actually accurately target metastatic disease, you actually give, in theory, the patient a much better chance of disease-free progression moving forward, which for this particular field is a huge leap, because the 1 to 5 year survival rate for women who present with high-grade serous ovarian cancer is actually very, very low. So any sort of progression-free survival that you can afford to these patients is extremely beneficial for this particular population. So the screening campaign that we undertook after the oncology-directed library of small molecules that we talked about was 100,000 compounds that comprised many different libraries. There was Citribon, we had some in-house libraries, we had some analogs that we were testing. And what actually came out of those was a cluster of compounds that had differential effects. And these clusters actually grouped together beautifully according to chemotype, and as you can see, there were various cutoffs that we had for the different clusters. We wanted to look at compounds that inhibited metastasis but had no effect on cell viability, because those were the ones that we thought would be great to use in conjunction or in combination with standard of care chemotherapeutics for ovarian cancer like paclitaxel and cisplatin to be able to prevent growth of the primary tumor as well as metastasis. In addition to that, you also have other clusters of chemotypes that prevented metastasis as well as killed the cells, and and this would be an all-in-one drug. So the idea was to be able to separate out these various different groups of compounds, if you will, and then investigate them further by doing medicinal chemistry and developing different classes of drugs. So having basically established then that the assay that would report on metastasis, as it were, we had another question. We said, well, how do these cells really metastasize to this metastatic nature of the amentum? Do they come as single cells? Do they come as spheroids? Do they come as aggregates of cells that are enriched for cancer-like stem cells? So for that, what we did was we actually ran proliferation assays on the omental layer that we had established on the three-dimensional omental layer, but had the incoming cancer cells as either 2D or 3D spheroids. And as you can see by the graphs, by the bubble graphs on the left, the microenvironment really does matter. It makes a huge difference in terms of what the morphology of the invading cell looks like. And as you can tell, there are certain classes of compounds that prevent the proliferation or the cell viability of these at the metastatic niche when they're proliferating as three-dimensional spheroids versus two-dimensional cells, which is not really realistic in an in vivo setting. So it really led us to a point where we could identify compounds and targets that are important intracellular, as for intra as well as intercellular communication in metastatic models. Having established the small molecule screen for this, we were interested in perturbing the microenvironment using functional genomics approaches. And to do that, what we did was we took the patient fibroblast and mesothelial layer and actually did an siRNA screen there, where we actually, and the data from here is from a microRNA screen that we did before we started the siRNA screen. But the idea was to see when you perturb the microenvironment by knocking down certain genes in that system, how does that affect the metastasis of the cancer cells through the system? And as you can see, we actually got some fairly interesting targets that we are now following up on. So the idea here was to develop an assay that could be used across various functional platforms to be able to not only identify a chemical modulator, but also to identify a target that would potentially go along with that modulator. Having established that we have this metastatic model, I wanted to talk to you a little bit about the initiative here to bioprint human tissues that's being led by Dr. Mark Farrar. In conjunction with Organovo, there are programs at the to basically look at architecturally defined tissues. And the idea is that this falls into part of these three-dimensional layered platforms where you can actually develop tissues that will inform on whether your three-dimensional assays were predictive and how far away in terms of prediction and prognostic value the 2D assays were from the three-dimensional assays. So to that point, when we come back to talking about what is a 3D microtissue and how the bioprinted tissue serves as a method to validate compounds that are coming out of these models, I'd like to draw your attention to the 3D tumor spheroid model, because there's a lot that's out in the literature about 3D tumor spheroid models, and we do believe that it's a question of what is your best fit for the biology that you're studying. Are you going to look at only single-cell tumor spheroids? Are you going to look at single-cell and multi-cell type tumor spheroids to try and mimic the stromal effect on the cancer cells? Do you have to throw inflammatory cells into this system? Does the size of a spheroid matter? These are all the questions that we seek to learn answers to in terms of developing screening models. And as you can see in the next slide here, what we really want to be able to recapitulate are really the pathophysiological gradients in tumors and 3D tumor spheroid models. So if you look at the graphic on the right, you can see that there are decreasing oxygen, nutrient, and pH gradients from the blood vessel, which is technically where your drug or small molecule is coming from, to the actual tissue where you have the solid tumor models. Now, given the fact that these gradients exist, it's a reasonable expectation to wonder, well, how much of my drug actually gets to the tissue? How many cells does it kill along the way? Do the gradients affect the potency of the drug? And as you can see on a spheroid on the left-hand side, there are actual zones within the spheroid that may or may not affect the potency and the efficacy of the drug in question. So for us, really, what was most interesting is, in terms of investigating, was the yellow quiescent zone, because this is the zone that is actually enriched in cancer stem-like cells, which are responsible for repopulating the tumor. So when you actually hit a tumor spheroid or a tumor, primary tumor with cytotoxic drugs, the first cells to go are the outer rapidly proliferating layer. And this is usually what completely disintegrates the tumor. However, it's the yellow zone where these cells are not that affected by the drug due to the gradients that we already discussed, but they actually have the ability to survive the cytotoxic insult and then mutate and morph into a different type of cell that can then repopulate a genetically different tumor. This is the population of cells that we seek to really eradicate through our 3D screening models. So to do that, we've set up a platform of cell-based assays in terms of increasing physiological relevance. The idea of this is to investigate the effect of target perturbation on cancer cells in model systems. So you have a two-dimensional layer, something that everyone's extremely familiar with, and then you have a 3D spheroid and a 3D organoid model. In terms of high content assays and high throughput screening, the 3D spheroid assays are those where you have spheroids from 30 to 50 microns, and you have multiple spheroids per well. In terms of what cancer biology that's mimicking in vivo, what it really is mimicking is the inability of the cells to attach to plastic and the signaling systems that are affected by this. But the 3D organoid models, which actually have one organoid per well, are the real indicators of what is happening to a drug or any sort of genomic perturbation in the context of pH gradients, nutrient gradients, and the cell layers that you have within. So in order to garner information about the difference between 2D and 3D systems, we typically run all assays against these three high throughput systems in increasing physiological relevance. And as you can see here, this is a snapshot of what we hope will be useful to the community. So the idea here is really to develop a blueprint, if you will, for each compound in terms of what it does to the endpoint, which is viability, but also what it does morphologically. And I think the data on this slide will make that point, drive that point home a little more clearly. So if you look at the two graphs on the left-hand side, we have dose response curves for seleninomycin and vorinostat in a model of high-grade serous ovarian cancer. And as you can see, they both have very similar IC50s, extremely potent at causing cell death. However, if you look at the graphic on the right-hand side, you can see that they actually have very different effects on the morphology of these organoids, even though the end result is the same. So if you look at the upper panel, which are organoids treated with seleninomycin, even at the highest concentrations, you find that the organoid is completely destroyed. However, if you look at vorinostat and look at the same morphological effect at the highest concentration, I draw your attention to the bright field pictures. You'll find that the organoid is still completely intact. Again, this goes back to our discussion on the appearance of cancer-like stem cells within a tumor and how they're able to repopulate a tumor, which is a genetic variant of its original type, when they're actually exposed to cytotoxic insult. Having established a platform where we can develop the blueprint for these small molecule compounds in a single cell type, we then moved on to seeing what the effect of a stromal cell would be on the cancer cells. And the reason to do this is to build in the complexity while moving towards a more relevant 3D screening model. Cancer cells do not exist as islands within the body. They're heavily entwined with the stromal counterparts, with the inflammatory counterparts, with the endothelial counterparts. So really, to be able to get to reach an answer, we have to be able to build in a certain degree of complexity that mirrors that biology in a human being, but also not to a point where the assay then does not become robust and reproducible. So that's kind of the gray area in screening, where you have to make some concessions in terms of how relevant you can get so that you can actually get an assay that is predictive. So the first thing that we did was, in conjunction with Spheroid, we developed a multi-cell type tumor model where we looked at HAE8, which is an ovarian cancer cell line, with and without with and without fibroblasts. And on the left-hand side, as you can see, you have staining for EGFR, Ki67, collagen, and as you can tell from comparing the staining profiles, when these cells are actually grown with fibroblasts or their stromal component in vivo, you have a much higher expression of many of the proteins like collagen, Ki67, and ECM. What's remarkable, though, in these multi-cell type tumor models is that there seems to be some sort of tissue memory or cellular memory within each cell type, because no matter how you mix the cells together, the epithelial cells will always form this outer ring-like cover around the fibroblast of the stromal or the mesenchymal stem cell core. And it seems to be some memory of the way that the cells associated with each other while they're in separate culture. When you actually compare the maximum responses in terms of drug responses, which you can see on the right between the homo and heterotypic tissues, you find a wide variation in terms of not only what their responses are, but also how long it takes to respond. And typically what we find that, and this is actually pretty intuitive, typically what we find is that it's not only a component of time, but it is the addition of these protective, if you will, stromal cells to the cancer cells that actually protect them or modulate their response to a particular chemotherapeutic. Having established that multi-cell type tumor models could potentially give us more answers about what targets are involved in a chemotherapeutic response and how you can modulate that to actually get a therapeutic window where you kill epithelial cells and not the stromal components so that you prevent systemic toxicity, we then took this assay to an RNAi screening platform where we performed RNAi high-throughput screening of both single as well as multi-cell type tumor spheroids, because the idea was to look at not only morphology but also viability in 2 and 3 dimensions. So breast cancer, this was a comprehensive analysis of breast and colorectal cancer in 2 and 3D, and then we did the colorectal cancer study with and without fibroblasts. And as you can see here on the next slide, we looked at the difference between breast cancer and colorectal cancer in 2D and in 3D. And we identified certain genetic targets that are specific only to the geometry. So these were things that were not necessarily different between breast and colorectal cancer, but they were certainly different with respect to what grew in 2D versus 3D. Furthermore, when we then took this and did an analysis between each cancer type, so targets that actually came up as positive for just breast cancer or just colorectal cancer, as you can see in figures C, D, and E, you can actually identify gene targets that, when knocked down, would very reproducibly cause the same differences in phenotype between 2 and 3D. And some of these gene targets were actually the same for both colorectal cancer as well as breast cancer. We then looked at the protective effect of the stromal component, the fibroblasts, in a model of colorectal cancer. And as you can see in panel A, those are the scatter plots for the viability readouts for this. And in B, you actually see the immunostaining of 2D versus 3D single cell type versus multi-cell type tumor model, wherein the knockdown of these specific genes actually ends up giving us a particular phenotype. And we were able to successfully identify and validate many of these gene targets as being differentially regulated in colorectal cancer in the presence and absence of stromal fibroblasts. Having established a viability as well as a morphological readout for colorectal and breast cancer, we then turned our attention to the equivalent of the small molecule screen for metastasis. Now, this was an assay that we actually developed for breast cancer, and the idea was to look at migration in 2D, which you can see on the left, and then invasion in 3D, where you actually have these 3-dimensional breast cancer organoids that will invade into matrigel, and you're looking for the knockdown or the silencing of certain genes that will actually prevent this effect, because those would be directly implicated in metastasis of the breast cancer cells. And this project is actually currently ongoing, but the idea is to use these assays to interrogate or identify really epigenetic targets of breast cancer migration, both from a gene silencing as well as a small molecule perspective, to be able to tie again a modulator to a target and vice versa. Having established the RNAi component of the functional genomics platform, we've done a couple of preliminary studies using CRISPR-derived cell lines to look at the effect of physiological relevance on targeted chemotherapeutics. So for this, we used a lung cancer model where we had CRISPR cell lines that were targeting the ALK mutation. And as you can see, once you have that genetic mutation built into the cell line, using a 3-dimensional organoid system gives you a huge shift in the responses, which you would expect to see in an in vivo system, whereas in the 2-dimensional system, there's no real change in the way that the responses shift. So to actually make even in a model where you have a genetic component, having a 3-dimensional model really gives you a sense of predicting further on how good your chemotherapeutic is going to be in terms of A, diminishing tumor size, but B, also giving you a therapeutic window. So with that being said, really, what I'd like to end with is the fact that physiologically relevant phenotypic assays such as those looking at three-dimensional systems, does really create a much more relevant model system for both target validation and drug discovery. And both target validation and drug discovery from a screening perspective should really go hand in hand, because it really helps identify new targets for existing drugs, and then on the flip side of it, deconvolve the polypharmacology of existing drugs for certain target classes. And the use of physiologically relevant assay platforms and the development of these assay technologies, such as those that we can do in three-dimensional multi-well pleats, is really key to unlocking and perturbing biological systems. One of the biggest lessons I've learned from working with a huge and multidisciplinary, diverse team is that translation truly is a team sport. There is no man is an island, no scientist is an island, and to be able to really think about disease in this context and accurately reproduce this in vitro so as to have better candidates that enter clinical trials, we truly do have to work as a multidisciplinary team to be able to bring this to fruition. So with that, I will close, and I'm happy to take any questions.
Excellent presentation, Dr. Lal-Nag. Thanks for bringing that information to us. So before we get started with all of your questions, here's a quick reminder about how to reach us today. Questions can be sent via the green Q&A button at the lower left of the presentation window. We'll get to as many as we can. Our first question is, do you see 3D models replacing animal models?
That's an excellent question, Judy. I would have to say the idea from a perspective of developing these screening models is not to replace them necessarily, but to be able to be as predictive, because the idea is we can screen hundreds and thousands of compounds in these assays, but you can't do the same experiment in 100,000 mice. So if we can get to a point where we're as predictive as an animal model, that's all we really need to go into a preclinical stage.
What are the minimum requirements for a 3D model to be relevant?
So that's a question we get a lot because many different collaborators, many different academicians are looking at different aspects of biology. So for example, if you looked at our breast cancer migration, it was really sufficient to be able to put breast cancer cells in Matrigel, because that's it really sufficiently recapitulates the stromal environment of breast tissue and look for metastasis. However, if you go back to when we were looking at the metastatic model for ovarian serous carcinoma, it gets much more complicated, because you have multiple cell types in various stages. So you have an amenable layer where you have cell type, 3 different cell types, and then you have an invading cancer cell. And they're all equally important in answering the biological question. So I think when it comes to 3-dimensional models, it really depends on the question that you're asking. If you're asking about chemotherapeutic interactions and the protective nature of a stroma, maybe just having 2 cell types is enough. But more and more with the development of immuno-oncology therapeutics, it is coming to our notice that having an inflammatory component, which basically allows the cancer cells to be protected from the immune system, is also an important component in these assays, which has to be somehow incorporated, because if you want to go to animal models and you have an immune system and a functional immune system in the animal model, you're not going to be able to do an apples-to-apples prediction if you're not sufficiently recapitulating that in a 3-dimensional model. So I really do think that it depends. The complexity, it's not a one-size-fits-all. You don't want to have all cell types for every 3D assay that you run. I think it's the minimum requirements for the biological question that you're answering for a particular assay.
How reproducible are the results of assays in 3D systems?
Surprisingly, and I would have actually thought they would be less reproducible than we find them to be. And even when you talk about, like, as I mentioned with the assay that we looked at for metastasis, when we were looking at cells from different patients in every assay run, the fact that it was so reproducible in terms of the target classes for compounds that it was returning was actually incredibly remarkable for us. So the one thing I will say about 3D systems is I think on some level the biology takes over, and the biology is so robust, or the biological question that you're asking in a 3D system is so precise that these assays tend to be pretty reproducible.
Thank you. If we didn't get to your question, we will follow up with you via email. I would like to once again thank Dr. Lal-Nag for her presentation. Do you have any final comments?
I would just say, I think, well, I'm a little biased here, but I think everybody should consider moving towards much more physiologically relevant models, because I do think from an entire industry perspective, we'll be doing both academia as well as pharma a huge favor.
Thank you again. I'd also like to thank our sponsor, PerkinElmer, for making it possible to bring this presentation to you. Today's webcast will be available for on-demand viewing through May 2018. You'll receive an email from us alerting you when it's available on demand and posted on labroots.com. You're welcome to forward this announcement to any colleagues who weren't able to join in today. Thanks for logging on and participating in today's broadcast. We'll see you next time.